Hello, everybody. My name's Anna, and my partner's name is Bianca. And the study we decided to look into was the complete mitochondrial genome and evolutionary analysis of Turritopsis dohrnii, the immortal jellyfish with a reversible life cycle. So, first things first, we wanted to hop into some background of Turritopsis dohrnii. Tissue and cell differentiation in most animals is irreversible. Once cells commit, they don't go back. However, the jellyfish T. dohrnii is exceptional because it can reverse its life cycle, transforming from an adult medusa back into a juvenile polyp when stressed. This transdifferentiation ability challenges the typical rules of development and makes T. dohrnii an important model for studying aging and cellular plasticity. While some genes from T. dohrnii have been sequenced previously, its complete mitochondrial genome is unknown. So as we know, the mitochondria is responsible for producing cellular energy and helps regulate stress responses and homeostasis. Mitochondrial DNA is widely used to study evolution because it reveals relationships among species and highlights metabolic adaptations. Given T. dohrnii's remarkable biology, determining its complete mitochondrial genome helps us understand its genetic features, compare it with related species, and explore how its mitochondrial DNA may relate to its regenerative abilities. The study aimed to comprehensively characterize the mitochondrial genome of T. dohrnii. The goals were to sequence the complete mitochondrial genome, describe its structure, genes, and repeat regions, identify species-specific mutations and selective pressures, predict how these mutations may affect mitochondrial respiratory proteins, determine its phylogenetic placement within hydrozoa, and assess genetic diversity across different populations. We chose this study because T. dohrnii is one of the only organisms known to naturally reverse its life cycle, making it biologically immortal. This phenomenon is rare and understudied, since the species offers a unique window into aging, rejuvenation, and mitochondrial biology. It's an exciting and unconventional model to explore. Alright, now jumping into the methods. So the researchers from this study collected T. dohrnii from two locations, Italy and Turkey, to compare these populations. To get enough material, they pulled several polyps and medusae from each site and extracted DNA. Samples were prepared for sequencing and run on an Illumina machine, which provided most of the data. For difficult regions, especially the duplicated COX-1 gene, they double-checked the sequence using traditional Sanger sequencing. Afterwards, they assembled all the sequencing reads into mitochondrial genomes. The Turkish population BP had very high coverage, so they were able to reconstruct a complete mitochondrial genome. The Italian sample, T10, was almost complete but was missing a small region. Alright, so once the mitochondrial genome was assembled, they identified all the genes using automated tools and confirmed them by comparing with known hydrozoan sequences. They refined the protein coding genes to make sure each gene had the correct reading frame. They also predicted all tRNA and rRNA genes. To understand how genetic differences might affect function, they modeled the 3D shapes of the mitochondrial proteins. Finally, they looked at the genome's overall composition, like which nucleotides are most common and how codons are used. These analyses mainly focused on the BP genome since it was the complete one. To understand where T. dohrnii fits among hydrozoans, they created a dataset that included mitochondrial genes from many related species. They excluded the duplicated COX-1 gene because it could bias the results. They aligned all gene sequences, cleaned the alignments, and combined 13 protein coding genes with one rRNA gene into a single dataset. Using this combined dataset, they built evolutionary trees with two different methods to make sure the results were reliable. They also tested whether any genes showed signs of selection and measured how much genetic diversity exists within T. dohrnii compared to other species.
Hi, I'm Bianca. Now we're moving into the results and discussion section of the article. On the right, you can see the predicted protein structures for NADH4 and NADH5, which are both components of complex 1 in the electron transport chain. If you look at NADH4 on the bottom, there's going to be black arrows showing an extra alpha helix and additional beta strands, and these extra structures aren't seen in related hydrozoans. For NADH5 up at the top, you're going to see a longer beta strand depicted by this black arrow, and it's going to change the overall shape of the protein's catalytic surface. So because complex 1 helps regulate ATP production and oxidative stress, even small changes can affect how cells handle damage. So since stress is what triggers the jellyfish's life cycle reversal, the modified structures may help T. dohrnii tolerate extreme stress and successfully reset back to its polyp stage. COX-1 is another mitochondrial gene that shows dramatic differences in T. dohrnii and may play an important role in its unusual biology. COX-1 is part of the complex 4 in the electron transport chain, which handles the final step of ATP production and regulates how cells cope with oxidative stress. The COX-1 protein in T. dohrnii is 52 amino acids longer than in related species, which is unusually high. So on top of that, a portion of the COX-1 gene is duplicated at the opposite end of the mitochondrial genome. And both these features suggest that COX-1 protein might fold differently or behave uniquely under stress. And because of the life cycle reversal that's triggered by damaging events like starvation, injury, or environmental changes, modifications to a stress-regulating gene like COX-1 could be meaningful. Another striking discovery in this study is the highly polymorphic G repeat region at the 5' end of the mitochondrial genome. So the repeat region shows about 67% heteroplasmy, which means that there's many different versions of the mitochondrial DNA sequence coexisting within the same animal. And this level of heteroplasmy isn't seen in other hydrozoans, and it suggests that there's a very dynamic mitochondrial genome that may shift or respond quickly to stress. So since the jellyfish's rejuvenation process happens when the organism is under stress, this flexible mitochondrial system might be supporting its ability to transition backward in development, like back to the polyp. And the authors don't yet know the function of the repeat region, but because it's so variable and so unique to T. dohrnii, it's a major direction for future studies. There were 4 mitochondrial genes that showed signals of positive selection: ATP8, NAD1, NAD2, and NAD5. These genes are part of the oxidative phosphorylation system and directly support ATP synthesis and proton pumping. Positive selection means that certain amino acids provided an evolutionary advantage and they were retained over time. So because these genes influence energy production and management of reactive oxygen species, selection here may reflect adaptations related to stress tolerance or metabolic efficiency. And this is especially interesting in the context of T. dohrnii because the species undergoes a massive metabolic and cellular shift when it reverts from a medusa back to a polyp. And these selected genes help support the energetic and stress-related demands of that transformation. So now we're looking at a figure from the article that compares synonymous and non-synonymous diversity across hydrozoans. If you look at the light blue line for T. dohrnii on the top graph, which represents synonymous diversity, you can see that its synonymous diversity is very high and up to about 20% in some genes, like right here. And these synonymous mutations don't change amino acid sequence, so they're usually neutral and accumulate over long periods. And the high levels indicate a large genetically diverse population. On the bottom graph down here labeled B is going to be the non-synonymous mutations, which do change amino acids, and it's going to show a much lower diversity, meaning that the most protein-changing mutations are constrained, and there are a few that persist though, like NADH4, NADH5, and COX-1, which were previously already mentioned as being important. And overall, this figure is just showing that T. dohrnii has unusually high genetic variability, which may support its ability to handle stress and undergo the life cycle reversal. So this is table 2 from the article, which compares mitochondrial genetic diversity among several hydrozoan species. And if you look at the row for T. dohrnii, you can see that its diversity is clearly elevated compared with species like Physalia or Ectopleura. And you can also see that it does, however, fall below Aurelia, and that's because Aurelia is a cryptic species complex, which means it actually represents multiple hidden species grouped under one name, which inflates its diversity. So it's expected that T. dohrnii is below them. So within single well-defined species, T. dohrnii ranks among the more genetically diverse hydrozoans, and that level of diversity supports this idea that the species has strong evolutionary adaptability, which fits well in turn with its ability to survive stress and reset its life cycle. So overall, the researchers found many key differences in the mitochondrial DNA of T. dohrnii comparatively to similar species, and hopefully these differences make it easier to find out why this is the only species that can revert from an adult to a juvenile.
Although this study provides the first complete mitochondrial genome of T. dohrnii, there are several important limitations we would like to acknowledge. First, this dataset includes only 2 populations, meaning it likely only captured a fraction of the species' true genetic diversity. The T10 genome was also incomplete, particularly around the duplicated COX-1 region and rRNL, which may affect comparisons. Because it focused solely on mitochondrial DNA, we're only seeing maternal inheritance, leaving out the nuclear genome, which carries most genes involved in regeneration and aging. Some findings, such as predicted structural changes in respiratory proteins, are based on computational modeling and will require more experimental validation. Additionally, the presence of highly polymorphic repeat regions and high heteroplasmy made assembly more challenging. Finally, phylogenetic interpretations are still constrained by the limited number of available hydrozoa mitochondrial genomes. Despite these challenges, the study opens several exciting directions for future research. For example, expanding sampling to more global T. dohrnii populations could help clarify its genetic structure and dispersal patterns. Incorporating nuclear genomes and transcriptomes could provide deeper insight into aging, cellular plasticity, and transdifferentiation. Laboratory studies could verify how species-specific mutations and structural protein changes affect mitochondrial functions. And as more hydrozoan genomes become available, we could refine phylogenetic relationships and get a better understanding of evolutionary patterns within the group. Overall, this mitochondrial genome lays foundational groundwork for studying immortality, regeneration, and evolutionary adaptation in T. dohrnii. Thank you so much for listening to our presentation, and we're open to any questions.